It certainly is so very appropriate to talk about conjugation when we're so near Halloween, because it's absolutely the most horrifying but yet the most fascinating of all of the lateral gene transfer processes. It certainly is the most important of the mechanisms by which plasmids get transferred. And as we turn our attention to the nuances behind this plasmid transfer, I want to begin by actually reviewing just a little bit of the historical context and a bit about the discovery of this sort of plasmid transfer. So I'm going to bring up a picture to talk about the experiments that were done, and based on our story that we told at the opening of our conversation about changes in genotype, recall that story about dysentery in post-World War II Japan. We had talked about how suddenly people began to realize that recombination and not just mutation was a method by which organisms could undergo change to their genotype, and this was, of course, in the mid-20th century. So experiments in 1946 by two gentlemen, Edward Tatum and Joshua Lederberg. These two gentlemen did two experiments in 1946 in which they actually were able to show that if you put in a beaker, and I'm just going to draw beside our other picture here, if you put triple auxotrophs, two different triple auxotrophs into a beaker together, remember that means that there are two different cells that each lack the ability. I'll make one cell green and maybe one cell blue, and each one of these cells lacks the ability to do three things. And what they found is that if they put these cells in together, then incubated them together, they would all become prototrophs. Holy cow. So what this meant is there was definitely genetic exchange taking place. Now, what Bernard Davis in the 1950s wanted to do was to find out the basic means, or the most important means, by which the plasmid transfer was taking place. And he wanted to prove that, in fact, cell-to-cell contact was involved. So he developed this ingenious experiment in which he put the cultures in a U-tube, not to be confused with like YouTube, but no, an actual U-shaped tube, right, where he would put one of the auxotrophs in one side of the tube, and he would put the other in the other side of the tube, and separating these two regions of liquid culture, he had a membrane. And this membrane has a pore size that would be large enough to allow naked DNA to transfer between the two sides, but not large enough to allow cell-to-cell contact between the cells, right? The bacterial cells themselves couldn't get through, so they couldn't make contact with one another. And what he found was that there was no reversion. There was no, these cells did not become prototrophic after incubation. So he was able to show, in fact, by this famous YouTube experiment, that cell-to-cell contact was necessary for plasmid transfer, thus discovering, in essence, if you will, the process of conjugation. So plasmids that are self-transmissible, we say that they are conjugative. So one of the most important of these plasmids is something called the F, or fertility plasmid. The fertility plasmid is quite a simple plasmid that really just encodes for its own transmission. So it's not carrying a boatload of other genes on it, but it does encode for what we call the TRA operon, which is transmission operon. It allows for the fertility plasmid to get from one cell to another. That is, it encodes for conjugation. So it codes for the structure of, obviously, the sex pilus, or the F pilus. Both terms describe the same structure. It contains the information for its own transfer, as I said, the TRA operon, which allows for the synthesis of all of those gene products that are needed for the F plasmid to get from a donor cell to a recipient cell. Now, this is where we always create everything in our own image, right? And we tend to use some terminology that's very egocentric. We call those cells that are donor cells and have the fertility plasmid, we sometimes call them male cells. We also sometimes call them F+ cells, and of course donor cells. So these are some of the names that those cells that contain the F plasmid by which they are called. Recipient cells, sometimes called female cells, do not contain the F plasmid. These are known then as F- cells. One thing that's super interesting about the fertility plasmid is that under some cases, it actually can, if it bears some similarity to the chromosomal DNA, it actually can integrate into the host cell chromosome. We thus term the fertility plasmid, we give it a special name, we call it an episome. An episome is able to integrate into the host cell chromosome. So you can imagine that sometimes you get a pretty crazy cell that actually has the transmission operon integrated into the chromosome, meaning that sometimes chromosomal DNA can be transferred, though that is not generally the case. So let's bear in mind that episomes can happen, maybe even highlight in your notes. But for now, I want to focus on what is going on when just the F plasmid in a donor cell is transferred from donor to recipient, the unidirectional transfer that takes place there. So plasmid transfer is always unidirectional. That is, it's not a situation of reciprocity. It's not a situation where the donor cell says, you know, hey dude, here's some DNA, take it, and then the recipient cell says, you know, woohoo, you know, thanks dude, I'm going to transfer back some other DNA to you. It doesn't work that way. The donor cell transfers the fertility plasmid to the recipient cell one direction, so F plus donor to F minus recipient cell. Donor cells transfer the F plasmid, but generally not their chromosomal DNA. Now, we'll talk about the exception to that, and that can occur when there's integration into the host cell chromosome and when the plasmid is an episome. But for the most part, when we're just talking about it being an F plus donor cell, regular old F plus donor cell, it's just transferring the plasmid, the F plasmid, and not the chromosomal DNA. Let's go ahead and look at this in a little more depth, and a picture is always the best way to do that. So in the very first step of conjugation, the first thing that has to happen clearly is contact, right? That's what Bernard Davis precluded, was that contact. Well, what will happen is that the donor cell will encode for the synthesis of the sex pilus. We can see that pilus forming here and going to be linking the donor cell to the recipient cell. So the F plus donor cell establishing contact with the F minus recipient cell. The two are now linked together. The next stage that is going to take place is called mobilization. Mobilization quite literally means that the F plasmid that's shown here in colors has to get cleaved at a site, and it actually gets cleaved at a site called the ORET. The ORET is such an appropriate name for this because it's the origin of transfer. So the origin of transfer, that's where the cleavage is going to take place. So a 3'5' phosphodiester bond is going to be broken right there in this step called mobilization. Now, once mobilization has taken place, then what will occur now is actually super fascinating. It's something called the rolling circle mechanism of replication. And I'm thinking that people like maybe Anna and Rochelle, Sarah, those of you who really like to bake are actually going to think this is a great analogy. My grandmother gave me an old apple peeler. I don't know if any of you guys have ever seen those. You like stick the apple onto these sharp spikes and then you set them on there and you like roll around this peeler and it pulls off the peel gradually. And this is kind of, every time I see that, I think rolling circle mechanism of replication. Because essentially what will happen here is as new DNA is synthesized, beginning at this open 3' hydroxyl, it will push, essentially, it will head this direction and peel off the existing strand that's shown in blue there. So it is like a peeling of the old strand of DNA as the new strand of DNA is made. So this is a very unique mechanism of replication, very different from the one that we have seen in the past. So here we watch the rolling circle mechanism happening. The old strand of DNA is peeled off, and in the steps called transfer and replication, we see the transfer of this one strand of DNA across this bridge to the recipient cell. Now, this is less well typified than you might think. We don't actually know whether it crosses across a hollow core in the sex pelvis or how that mechanism occurs, but we do know that it somehow gets across to the recipient cell, where it can then serve as template for the synthesis of a new strand of DNA that, of course, is going to look the same as the red strand in the original F+ cell. So please do note that the green strand of DNA is exactly the same as the blue strand of DNA, but I'm just showing it as something, a different color, so that you can recognize that it is the newly synthesized strand in the donor cell. So now a strand that's complementary to the transferred strand is synthesized in the recipient cell, and check it out. Here's what's going on. Now the F- recipient cell becomes an F+ donor cell. This is like a bacterial cell sex change, isn't it? It goes from being a female recipient cell to being a donor F+ cell. Marvelous, right? Now, this is crazy when you think about the rapidity with which this can take place. For example, if you take a 5 milliliter overnight culture and you put one F+ donor cell in that culture, it will turn the entire culture overnight to F+ donor cells. Absolutely nuts. But you know what else is cool? We've been situating our discussion a lot in E. coli, but it turns out that E. coli is not the only conjugative cell. And in fact, E. coli, now this is where we really get Halloweenish and sort of a horror story. E. coli can conjugate with not just other E. coli cells, but E. coli can also conjugate with Shigella and Salmonella. Interestingly, it can't conjugate with Proteus and Enterobacter, so it's not a family thing, but there certainly is a cross-genus conjugation that can take place. Now, before we leave this slide behind, I want to label the stages that we talked about. Number 1, contact. I used to love that show, 3, 2, 1, contact. I don't, for some reason that just went through my head. Contact is step 2, mobilization. And then finally, step 3 is replication and transfer. I put them together rather than having them be step 3 and 4 because they kind of happen at the same time. So we would understand those as being sort of the first step, the second step, and finally the third step in all of this process. Let's take a quick look at a wicked cool picture where you can actually see the sex pelvis of an E. coli cell. Check that out. Holy cow. So really this gives us an idea for the length of a sex pelvis. It can be particularly long. And we recognize that this could actually be occurring between two different genera, right? A Shigella cell and an E. coli cell. So we can see that cross-genus transfer. And you know what's really cool is this morning, actually on my Twitter feed, when we started our conversation of this, remember how we talked about the woman who post-mortem, all of her normal flora bacteria had become antibiotic resistant, and that primary means of transfer of the plasmid, the antibiotic resistant plasmid, is through conjugation. So in environments that are fraught with selective pressure, we see a great amount of conjugative transfer of genes. And this is why when we were talking about the use of the prophylactic use, that is the use of antibiotics as prevention. Remember we were talking about that in feedlot environments. Well, it turns out that feedlots are not the only place in which this is happening. This is an article just this morning on my Twitter feed. Legacy of decades of antibiotic use. Honeybees carry many different resistance genes. And what they're finding is because of the overuse of oxytetracycline as a preventative in honeybees, these honeybees, their normal flora, their normal gut flora are also resistant. So we have pressured them to transfer via conjugation antibiotic resistance genes. And these honeybees are fraught with resistance. All of their normal flora are resistant as well. So this is a great article to find, if you would, in the news. And what's fascinating is that in countries where where the use of oxytetracycline in honeybee farms has been outlawed, places like the Czech Republic and New Zealand and Switzerland, we don't see the abundance of tetracycline resistance genes. Holy cow! Answers in the question, isn't it? Let's go on and talk about the epic R plasmids. So F plasmids are just conjugative, right? They aren't complex. They don't encode for a multiplicity of genes on them. They just encode for their own transfer, their own conjugation. But R plasmids, they are the Halloween story. They are the ones that encode for many resistance genes, generally between 7 and 8 resistance genes sitting on a single R plasmid. So they allow for the resistance of everything from antibiotics all the way to things like heavy metals. Sometimes these R plasmids can have resistance to things, besides antibiotics, maybe things like Clorox wet wipes, right? So as many as 8 resistance genes on an R plasmid. They are conjugative. They have that operon that it encodes for their own transfer from a donor to a recipient cell. They probably result from transposons, right? The jumping genes jumping in, carrying antibiotic resistant genes, the complex transposons jumping into plasmids and jumping in perhaps to F plasmids, right? And carrying with them antibiotic resistance genes. One thing that's interesting about R plasmids is that unlike simple F plasmids, they are not usually episomes. So I didn't use the technical term there. You could jot that also into your note. They're not usually integrated. We could say not usually episomes. So unlike F plasmids, they don't tend to integrate into the chromosome, but certainly they do are conjugative, so they are able to encode for their own transfer. So other conjugative plasmids may contain other genes that maybe encode for a selective advantage. Synthesis of things like bacteriocins and other virulence factors, things that allow a cell to compete in any given environment, or allow them, as I think it was Carl who brought up the other day, to increase their ability to degrade unique toxic chemicals or unique sources of energy in unique environmental niches. So very interesting, all of the things that we can see on conjugative plasmids. Let's go back to this story of the idea that the F plasmid can sometimes become integrated into the host cell chromosome. When that happens and we find the F plasmid inside of the chromosome, it creates a very special type of cell. Maybe you've even heard of this cell. It's called a high frequency of recombination cell. And in an HFR cell, it actually is capable of transferring certain of its chromosomal genes. Because what will happen is that the ORI-T, the origin of transfer on the F plasmid, is midway through that F plasmid. And so the F plasmid will begin to roll off like the rolling circle mechanism, and some of the chromosomal genes will be transferred to the recipient cell. Now, never are all of them transferred. That would entail too long of a contact. So before the entire chromosome can be transferred, the recipient and donor cells separate. It would take like 100 minutes before all of the chromosome could be transferred. And never does contact stay established for that long of a period of time. So this means something really important. It means that never is the entirety of the F plasmid transferred. So unlike the regular conjugation between an F plus and an F minus cell, where after the transfer the F minus cell becomes F plus, in chromosomal transfer with an HFR cell, the recipient cell stays F minus. It doesn't ever get the entire F plasmid. So we see the transfer of certain genes, certain bacterial genes from donor to recipient, but we don't see that bacterial cell sex change, right? We don't see it going from being female to being male. A picture says 1,000 words, and what we see here's the integrated F plasmid shown in red. This is an HFR cell because that F plasmid has integrated. Here we see the rolling circle mechanism midway through, and you can see how part of the F plasmid has been transferred, but not all of it. And part of what is transferred are certainly bacterial chromosomal DNA. So certain genes from the donor cell are getting transferred. Now, the HFR segment that is transferred from the donor to the recipient may then integrate into the recipient cell, making a stable recombinant. But the recipient cell will remain F minus because never did the entirety of the F plasmid get transferred into that recipient cell. So this is a pretty sweet deal, amazing mechanisms by which cells can undergo changes to their genotype, And I want to end with a question for you. So this mechanism of lateral gene transfer can result in non-reciprocal general recombination and does not involve formation of a defective phage, so no formation of a defective phage, and no cell-to-cell contact. So this effectively rules out transduction, can't be transduction, can't be conjugation. But if it is a mechanism of lateral gene transfer that results in non-reciprocal general recombination, that sure sounds a lot like transformation. Have a wonderful weekend, and I hope that I see you all with your awesome posters ready to go on Monday. Let me know if you have questions on those, and just have a great weekend of study.